Okay, 7:05 now. So wait a couple of minutes before we actually get started. But just to say welcome to everybody who's joined so far, and just bear with us a couple more minutes. We'll just wait for late comments for a couple more minutes. So we've just got a question in the chat, so I'll just reply to everybody. Feel free to have your camera off or on, either way is fine. One thing I would say is that the connection can get sometimes a little bit iffy if everyone's got their cameras on. If that does start happening, then maybe turn off the cameras would be great. So me and our panel will have our cameras on throughout the talk. And just to remind you that throughout the talk, feel free to put your questions, comments into the chat, and then I can put them to our panel to keep that working smoothly. So we haven't had any new people admitted to the waiting room for a couple of minutes, so I think I'll get started. So welcome everybody to this ScotSci event, COVID vaccines, Ask a Scientist. You're very welcome this evening. So hopefully in this session, you will have an opportunity to put your questions, which I'm sure I definitely have plenty, and I'm sure you do too, to three experts in slightly different fields, all around immunology, infection, vaccines, and COVID. So I'll just introduce you now to our panel for this evening. So we have Professor Emma Thompson, who is Professor of Infectious Diseases at the MRC Centre for Viral Research at the University of Glasgow. We've got Dr. Thomas Christie Williams, who's a clinical lecturer at the MRC Institute for Genetics and Cancer at the University of Edinburgh. And we've got Dr. Megan McLeod, who's a senior lecturer in immunology at the Institute for Infection, Immunity and Inflammation at the University of Glasgow. So thank you to our panel for joining us tonight, and I think we'll get started with the questions. So starting out quite simple, maybe this one would be best for Megan to answer. How does a vaccine stop you getting sick in the future?
That's a really nice question, and basically my favorite topic, which is on the science of immunological memory. And what a vaccine contains is usually a small part of the virus or the bacteria that you're getting protected against. And it is injected usually into your arm, and it stimulates your immune system to make a specific immune response against that small part of that pathogen. So the COVID vaccines have a little bit of the SARS-CoV-2 virus in them. And when it goes into your body, your cells produce that small part, usually a protein, and your immune cells see that protein. And the mechanics are that there are cells called T cells and B cells that will recognize that small part. They will make initial immune response, and that's often why you feel a bit sick after a vaccine, because you do have an immune response to the vaccine. And then some of those cells will remain in your body, sometimes for a few years, sometimes for the rest of your entire life. And they will continue to survey through your body, looking for that vaccine, the virus coming back. And then they're able to make a really fast immune response because they were educated the first time around by the vaccine and able to give you this really tailored protective immune response if you meet the virus or the bacteria in real life. Hopefully that answers the question.
Yeah, that's great. So I've heard in the news, lots of people have been saying that this is a new type of vaccine, mRNA vaccine. So how exactly is the COVID or the various COVID vaccine, how are they new? What does that mRNA, what does that mean? And what's it different to other ones?
So in the past, most of the vaccines that we've given to people have had a protein. So if you've ever had the, let me think of an example, the hepatitis vaccine, that's got a bit of a protein of hepatitis virus in it. Or if you've ever had the polio vaccine that you've swallowed, that's the actual virus. So these are either protein parts of the virus or the actual virus itself. The mRNA is the genetic code of the virus. So it's not the actual virus, it's just a message that turns into the protein that you make an immune response against. And the mRNA gets into your cells after you're injected in the arm, and your own cells will turn that message into a protein, and then your immune cells will see that protein and make an immune response against it. So we're kind of taking a step back from what we have been injecting into people. And I think I was a little doubtful about how effective the mRNA vaccines would be, because it's a very new way of making vaccines, but they've been much more effective than we could have guessed. And I think are more effective than many of the vaccines that we've used in the past.
Okay, that's interesting that you're saying that they're more effective. Do you think that is something that we might use for other things in the future then?
I will not be surprised if we see the next generation of influenza virus vaccines, for example, coming out as mRNA rather than another protein-based vaccine that is used at the moment.
That's really interesting. Okay, so maybe this is one to aim more at Emma. So in terms of the vaccine dose, a lot of people have been saying that if you've had COVID, then you only need one dose, that you get a lot of the immunity from the first dose. So can you maybe just speak us through the 2 doses, whether the 2 doses are necessary?
Yeah, I can talk about that. Can I make a quick comment on the mRNA vaccines as well? Because I think it's really extraordinary. And really what we're doing with the mRNA vaccines is instead of injecting the protein, just like Megan was saying, we're putting the recipe into someone's arm. And so you're really moving the stage of manufacture from big factories in around us into our own cells, into the arm. So you're giving the recipe into the cells in your arm, and then those produce the protein, and then you raise an immune response against that protein. And the thing, one of the sort of really extraordinary things about that is that literally now we can pretty much, so we do a lot of genetic sequencing in the UK, and you can actually look at the sequence of the virus circulating, and then you can say, okay, we want to produce this vaccine, and literally print it. With the genetic code, and then inject it in, and so it's really going to revolutionize vaccines. It's extraordinary. And the other thing, I guess the other point that I wanted to make was that it's going to be much faster. So like, you can see how much more quickly we can produce vaccines now, because we don't have to try to produce vaccines that have to be formulated in a very specific way as proteins now. We can ask our bodies to do it for us. So and I was also surprised about how well those mRNA vaccines have worked. It's incredible, and I'm sure we'll see vaccines against lots of. I think this is going to be a new era of vaccinology, actually, and it will revolutionize vaccines and the availability of vaccines for other illnesses. And I would hope to see that for nasty illnesses that you see in low-income countries, for example. Including things like nasty hemorrhagic fevers and that sort of thing, but also flu, just like Megan says. And then right now your question was about vaccine doses. So yes, so most of the vaccines that are in trials at the moment, they actually all have the same sort of common pathway of producing a protein at the end, which is the spike protein. Some of them also involve other proteins that come from the virus as well, but the majority of they all involve this spike protein, which is that bit of protein that sticks out of the virus that you see swirling around on your screens on the BBC all the time. And so we're just learning at the moment now about how to quantify the immune response to that spike protein, and whether or not people are likely to need more or less doses. So one of the things that's becoming, so most of the vaccines that give 2 doses of one type of spike variant. It's quite likely, and you probably heard some conversations about this in the news and so on, that we may need to update with boosters in the future, possibly on an annual basis, because the spike proteins are changing shape. So, and that may become important, and it's certainly possible that we may need to give additional doses of vaccines to people for that reason. But your question was more about, was also about people who've had COVID-19 and who've getting vaccines. And so there's increasing evidence now that it may be the case that people who've had COVID-19 only need a single dose of vaccine. That isn't currently the official advice. So official advice at the moment would be to have 2 doses of vaccine, but that may change. And we certainly can see that people who've had a single dose post known infection, they produce a very strong immune response because you've been primed with you've had the infection first, and then you get a single dose of vaccine will then really boost the immunity. So it is very likely that the advice in the future will be that it might be sufficient to have 1 dose rather than 2 if you've had COVID-19.
So you touched then. Oh, go on.
Just to add, although I don't think there's any concern that having the 2 doses will have a negative impact, just that it may not be necessary. The immune system is really fantastic, that it just gets better and better the more times it sees a particular virus or a particular vaccine. So I don't think people should be concerned about having a second dose, even if they've had COVID in the past.
That's really good to know. Kind of like when they tell you to revise for exams, like keep coming back to it.
Yeah, I mean, we should say that there are lots of people that have had COVID-19 that were in the trials, for example, and then who were given 2 doses, and there's no suggestion that that's harmful. Right. It just might be more efficient in the future in terms of rollout of vaccines, just to give a single dose.
Yeah, that makes sense. So I guess you touched on it there, and it's a question that's on a lot of people's minds, is that the mRNA vaccine was much quicker to produce and could be a much quicker way of doing vaccines in the future. So I guess a lot of people are asking, okay, so how come it was so much quicker? And I think you kind of touched on it there, in that we're not producing the protein, we're making the body produce the protein. But is there anything else that you wanted to add in terms of answering that question?
Maybe Thomas would like to.
I can add one thing as a bit of background for the COVID vaccine. It was actually based on a piece of basic biochemistry done by a researcher in the States called Jason McClellan, who's an RSV researcher. And he looked at a protein in RSV, which is respiratory syncytial virus. It's very similar to SARS-CoV. And he found that the F protein in RSV has got 2 conformations, before it binds and after it binds, which is similar to COVID. It changes after it binds. And he found out that the pre-fusion conformation is much more immunogenic. That means it's much more likely to elicit an immune response. And he found out that if you tinkered with the genetic code of that protein, you could maintain it in the pre-fusion state, so in a more immunogenic state. And that insight they took into MERS, which is Middle Eastern Respiratory Syndrome, which is another coronavirus. And they designed mRNA type vaccine, or the structure of it, for the protein in MERS. So when COVID came along, SARS-CoV-2, they knew that if they tinkered with that code, they'd produce a spike protein that had a different conformation and would stay in that conformation and be much more immunogenic. So that's partly the reason it's been so effective, is learning from previous viruses.
So when we say that the mRNA vaccine is new, actually the science behind it seems to have been going on for quite a while.
Yes,
yeah, that's right.
Yes, it's been going for 20, 30 years, people trying to develop it.
Wow. Okay, so it was kind of like a right time, right place situation, in that they were looking at another type of coronavirus just at the moment that we needed a really quick turnaround for a new one. That's really great. So Thomas, I know that you're interested in genetic variability, and I think Emma kind of mentioned there about boosters. Would that be because of these variants that are happening? We know that these variants seem to be happening in the spike protein. So is that why we would need boosters?
Yeah, so that's a really interesting question. I'm interested to hear Emma and Megan's views on this. So most vaccines are based on the spike protein. either directly giving your body a protein, a spike protein to develop an immune response to, or getting your own body to make that spike protein for itself. And then we know that coronavirus mutates over time just because all RNA viruses mutate, they change their genetic code. And that's probably mainly due to the machinery that replicates a virus, just randomly introduces errors. So if you have lots of virus growth and new generations of viruses, they'll make errors, and some of them will be carried on into future generations. As Emma was saying, I think at the start of the pandemic, I don't think anyone would have predicted that the mutations would have much of an impact on how the viruses behaved. But it turns out that they have quite a big impact, and they impact how the virus transmits. They impact how the virus affects people in terms of disease, and they also affect how the virus responds, or how your immune response responds to the virus. So it turns out that there's some mutations in that virus, changes in the virus, that mean that the antibodies you produced to a similar virus are less effective. So all the mRNA or the COVID vaccines are based on the original Wuhan sequence, which was the sequence from China back in January 2020. And then since then, we've had a lot of change, a lot of new variants. And you'll have heard about the Kent variant, or the South African variant, or the Brazilian variant. And they're all different strains of SARS-CoV-2 with genetic changes in that spike protein. And it seems as if those changes affect the way in which your antibodies you made to the virus can deal with that virus. So everyone out there today who's had a vaccine or is about to get a vaccine will have had a vaccine based on the Wuhan original sequence. But that means that our antibodies will be matched to that original sequence rather than these new variants. And we're seeing more and more that specific changes in the spike protein means that those antibodies bind less effectively. However, the good news is, I think Megan will hopefully back me up, is that even though your antibodies might not bind so effectively, your T cells, which are what protect you against severe disease, they're pretty clever. They bind to the virus in a larger number of ways than the antibodies, and it seems that your T cell response is more robust, which is why, even though for example, the AstraZeneca vaccine doesn't protect so well against infection with the new variants, it's likely that they'll still protect you against severe disease, whatever the variant. So even if there's a lot of variability in the spike protein, I'm pretty hopeful and confident. I obviously see what Emma and Megan think, that we'll still be protected against severe disease with all these different new variants. It's just a question of getting to the point where everyone's vaccinated so they can be protected against severe disease.
Did you have anything you wanted to add to that, Megan?
I very much agree with what Thomas is saying. The best type of protection is that antibody response. If it can stop the virus getting inside your cells, then it's the most immediate defense, because it can be immediately in your blood or in your mucosa to protect you against infection. Your T cells, they need a little bit of a kick to get going, so they can't stop you from being infected, but they can absolutely help to control the virus when you get infected with it. So that's this difference between stopping infection versus stopping severe disease. And it would be fantastic if we had viruses that didn't mutate, and then we wouldn't have this problem. But we see this every year with influenza virus, and we are reasonably good at managing that. And I think we can be better at managing that, because people like Emma and Thomas are tracking COVID-19 incredibly detailed way. And also we have these new viral platforms, both the mRNA and the viral vector ones, that we can more rapidly update to the latest types of virus strains that are around. So we can get those booster shots into production and into people's arms much more rapidly than we in the past have been able to do with the flu vaccine, for example. Emma, do you want to add to that?
Yeah, I mean, I might just say that we were really surprised by the new variants when they arrived. We watched the virus sort of changing shape, maybe with just 2 changes a month or something like that for several months between at the very beginning of 2020, when we started to import cases of COVID-19 into the UK, and so the virus was actually not changing very rapidly at all. And then suddenly in September and October, to the end of 2020, we suddenly saw these variants of the virus which had multiple changes in them. And these are the new variants. And a lot of those changes were in spike, although also there were changes in other genes or proteins in the virus as well. And I noticed just I guess I just noticed a question in the chat there, so it might just be worth saying that some of those changes are to avoid antibodies, and we think that that these variants arose in areas of very high transmission where the virus was under pressure because some people already in the population already had antibodies, and so the virus was finding a need to change shape to evade those public antibodies, like meaning that antibody responses that were common to several members of the population in these high transmission areas. But there were other changes as well. So and someone's just brought it up in the chat about why some of the variants were more transmissible. And actually, so B117, for example, which is the variant which originated in Kent, and has now spread globally and become dominant in many countries, it actually is able to bind more efficiently to the ACE2 receptor, so in the lungs. So you have this sort of interaction between the virus and the receptor, which sits in the lungs. And in fact, some of the changes within these new variants allow the virus to stick. It becomes more sticky. And the technical term is that it increases the binding affinity. But the virus actually sticks more easily to your lung cells, and we now know, unfortunately, that actually makes the illness a little bit worse as well. And there's increasing evidence that the B117 virus is actually a bit more difficult to deal with. It's a little bit more tricky in terms of your likelihood of needing oxygen, for example, or having to be admitted to hospital is higher with the B117 new variant. And then, but it is very sensitive still to vaccines, which is really good news. But there are other variants, in particular one, which is the B1351 South Africa variant, which seems to be associated with a slightly less effective immune response. So it's still very definitely worth being vaccinated, even if you're very, in South Africa, for example, where that variant has been quite dominant. But the likelihood of the vaccine completely preventing an infection or preventing you from having a serious illness is a little bit lower. And so it's likely in the future, because of variants like that we may want to update the vaccines. So I guess that's the... Yeah, so the new variants, they vary in two ways, and one is to escape immunity, but the other is actually they can make the virus a bit more sticky and more transmissible as well.
Great, and you've answered my next question, which was how are some of the variants more transmissible, and you've just explained that quite clearly. So we've got a question which kind of goes back to what you were saying, Megan, about the B cells and the T cells, and it's about age. So how do your T cells, the question specifically asks, but also I guess generally your immune system, how does that alter with age, and also how will the vaccine have an effect, like during age? Obviously a lot of the trials have been done in adults, but we know that older people get more sick and are more likely to obviously get severe COVID-19.
Yeah, like many things in your life, your immune system gets less good at protecting you as you get older. Your T cells come from an organ called the thymus, which sits just above your heart, and it actually starts to not quite shut down, but stops working quite so well, even in your teenage years. So you've already reached your peak. Probably most people in this call have reached their peak T cell. That's not to say you don't have new T cells coming out, it's just fewer of them. And your B cells also, they come from your bone marrow, and they also start to decline as you get older. But the fantastic news with the COVID-19 vaccines is that they induce a really great immune response in elderly people. So when they've compared the immune responses in middle-aged and young adults to more elderly people in the original trials, the immune responses were equally large. So that gives us really great confidence that these vaccines are protecting the elderly. And I think we're starting to see that in the data in the UK, that the numbers of people going into hospital and the deaths are reducing. There was a story in the BBC News this morning that seemed that there's some data to suggest we're seeing the impact of the vaccine protecting the elderly people who were vaccinated earlier at the end of last year and this year. So I think that's one of the things I'm really excited about the new vaccines, that they can give us this nice large immune response even in elderly people. Yeah. So there's hope even as we all age.
Even past our teens into old age. Okay, so great. I think we've covered now a lot of like how the vaccine works, how it's different, how the doses work. So we're getting quite a lot of questions, as you might imagine, about side effects. So I'm just going to start with a maybe easier one, which is that there's been lots of anecdotal evidence, although maybe you can inform me of some actual trials that have been done about people who've had COVID-19, either were symptomatic or were tested positive, and now they've had their first dose of their vaccine and they seem to have had a worse side effect profile than negative or people who haven't had COVID-19. And why is this happening?
Megan, you seem best placed, I'd say, to answer that.
Oh, I feel worse placed. I'm the only non-clinician here. It may be that you have, so if you've had COVID previously, you'll have antibodies and T cells and B cells that recognize the virus. So they may get that early boost when you get the vaccine, and there will be an immune response. So you will get those flu-like symptoms from release of soluble molecules that change the way your brain feels a little bit and makes you feel tired and then maybe a little bit sick. And so that's normal. Because it's immune response that's happening, and for the vast majority of people that will be relatively soon after the vaccine, and then it will clear up quite quickly. I'm going to make either of the two clinicians talk about whether that is anecdotal or really a finding that people have.
Emma, I know that you've been involved in some trials. I guess probably some of the people in the trials have had COVID-19 before they had the vaccine, so maybe you can give us some insight into that.
Yeah, so we're running 3 trials, 3 of the vaccine trials here in Glasgow, and then of course many of our staff as well who work for the NHS have been vaccinated, or most of us by now have been vaccinated at least with 1 dose. So yeah, there haven't been any sort of safety signals in terms of in these trials in terms of the likelihood of people developing severe side effects having had previous COVID-19. And actually that was one thing that we were a little bit worried about when we consented people for that quite carefully who were good enough to take part in the vaccine trials. And I'd really like to say thank you to anyone on this call who took part in a vaccine trial, because without you we wouldn't be anywhere. And we really rely on very large numbers of participants to come forward and take part in these trials, and that's what's made it possible for us to vaccinate people who are very much very vulnerable to the infection. So we're actually a little bit worried about certain types of immune response that might cause people who had previous infection to have a bad response to the vaccine, and actually the other way around as well. So if you had the vaccine and then became infected, that this might in some way train your immune system in the wrong direction. But actually that hasn't happened, and there's been absolutely no evidence of that at all, which is extremely reassuring. And but in terms of more sort of perhaps milder side effects that we worry a little bit less about, but they're still very important to people who are being vaccinated, and it may interrupt their day or the couple of days after they've been vaccinated. So I think it's very clear that if you've had COVID-19 already and you're vaccinated, you're probably more likely to be a little bit more likely to be fatigued, for example, and perhaps might have a mild fever and so on. But these wouldn't be expected to be problematic side effects, and which should be fairly easily manageable. But yes, I think there is reasonable evidence now that if you've had COVID-19 and you're vaccinated, that you might be more likely to get side effects, but not to any significant level that we would get very worried about vaccinating. And in fact, indeed, it's a good idea to be vaccinated if you've had COVID-19, because 1 exposure probably isn't enough. It's a bit like having 1 dose of vaccine. It's pretty good, and it does protect you to a certain extent, but 2 doses is often better than 1.
Great. Okay, so I guess that kind of goes back to what we were saying earlier about if you've had COVID-19, maybe you would only need 1 dose, but no harm in getting the second one as well. Okay, so along the same lines of side effects, so we've had a question about people who are on immunosuppressive drugs, or there could be various reasons for this. For example, if you had an implant, a transplant, not an implant, or various reasons. So how does that work? Because obviously the immune system is being repressed for good reason, but then that's exactly what we need from the vaccine. So what exactly is the advice that's been given around that?
Is that one coming to me?
You can go first.
Yeah, so the issue with immunosuppression of any kind is that you might not generate quite such a strong immune response, and we know, for example, from other vaccines in people who are immunosuppressed for various reasons, for example, with HIV infection, that it can be better to give a higher dose of vaccine. For example, for hepatitis B, we give a higher dose of vaccine, and so it can be better in certain cases to think about that, to try to think about ways to induce a better immune response. At the moment, there's a bit of we're in an information lag with regard to immunosuppression. What I definitely wouldn't suggest is, so usually people are on immunosuppressive medicines for very good reason. And so the question, I think, really was more about the kind of injected treatments that we give to people, for example, with arthritis or who might have had a transplant and that sort of thing. And so we definitely, I think it would be clinically pretty risky to suggest to stop immunosuppressive treatment before a vaccine, and unless that was something that was going to happen anyway, for someone whose arthritis was well controlled and so on. So it will be a case of discussing with your clinician, if this is you, about the pros and cons of what treatment you're on and the timing of vaccination. But generally speaking, at the moment, the recommendation would be for a routine vaccination, but discussion with your clinician. It may be, though, in the future that we find, for example, that giving an extra dose of vaccine or a higher dose might be helpful, and we don't know, we don't have the answers to that yet, but that information could change, but at the moment, in general, so none of these vaccines are live vaccines, so they're not likely to harm you if you are immunosuppressed, which is good news, right? Yeah. So because there are certain vaccines that we give which could be harmful and wouldn't be recommended in people who are immunosuppressed, but that's not the case with these vaccines. So it's just that the immune response might not be quite as strong as we would have hoped for, but we would, in general, still recommend vaccination. But I wouldn't just put a broad brush. I think because individual cases with different levels of immunosuppression and different reasons for immunosuppression should really be discussed with the specialists.
Yeah, absolutely. I guess this is probably a good point for me to say something which I meant to say at the beginning and then forgot in the excitement of it all, which is that obviously this evening we're trying to answer questions, discuss what's going on, which is a current event. Things are literally changing every day. And so for that reason, we don't know all the answers. Our lovely panel here, as much expertise and experience that they have, they don't know all of the answers either. And so they're giving their opinions today, which we hope that they're helpful. But of course, things might change. There might be a new development, or we might find out something new about how the virus works and how it evolves. And so yeah, a little get out of jail free card for our panel, that of course they can't know all of the answers because this is still a new and developing situation. And also, obviously, as Emma just said, don't be taking anything that we've spoken about tonight as medical advice. Obviously, if you're in a specific circumstance, then you should absolutely be speaking to your doctor, and they will be able to advise you in your personal circumstance. Okay, so having said that, obviously, as I'm sure you all expected, we've got quite a lot of people asking about the blood clots. So I guess the first question to ask is, I'm not sure exactly who wants to be the brave person to tackle this first, but what exactly are these blood clots? You know, people think, okay, blood clots, that's like when you get a scab, your blood gets thick, and then, you know, obviously that's bad if it's in a small vessel or in your brain, but it seems like people are talking on the news that these are a very specific type of blood clot, very rare, and they seem a bit different to a standard blood clot. So can anyone jump up and say, explain how exactly they're different?
Well, I start, and then, because I think Megan, for example, and Thomas might have quite a lot to add to this, but
yeah.
So the blood clots that seem to be possibly associated with the AstraZeneca vaccine are very rare. So like we're talking about events that are extremely rare. And then the second thing that's we should note really is that it's not just blood clots. It seems to be blood clots in association with very low platelets or thrombocytopenia. So it's a very specific syndrome of, very strangely, having very low platelets, which are the cells in your body which are involved in blood clotting. And they go really low, they become quite low, but at the same time you might develop a blood clot. And for example, people have developed blood clots in the cavernous sinuses, which are in your head, and it's around the brain. And anyway, the issue is that there seems to be a very sort of low level of extremely rare occurrence of blood clots in association with low platelets. And actually also it's been found that many people who've got this sort of syndrome have anti-PL4 antibodies, which maybe Megan knows more about than me. It's definitely not my area of expertise, but these are antibodies that are very specific and quite kind of unusual. So it's not like the kind of blood clots that people do get, and you know, maybe 1 in 1,000 people might get a blood clot anyway. And we find, so one thing to say very clearly is that the incidence of blood clots in the trials, when you look at the AstraZeneca vaccine, is much lower in people who are vaccinated against people who were not vaccinated. And the reason for that is that COVID-19 can cause blood clots. And so people who became infected with COVID-19 can have had blood clots. However, this particular syndrome with the low platelets and blood in association with blood clots appears to be occurring more frequently now that the vaccine has been kind of rolled out to millions of people. We are able to see these extremely rare events that we wouldn't have been able to see in clinical trials where perhaps 30,000 people were given the vaccine. And so once you get up into the sort of millions of people, it is possible to pick up very rare side effects. And I think this is actually, so it's concerning for everyone, but just to put it in some perspective, that you know, possibly these, you know, very rare side effects can occur with lots of medicines that we use, and actually we might be much less able to pick them up because we're doing such intensive monitoring at the moment that something that's incredibly rare actually does show up. And so the discussion around AstraZeneca has actually been because, so for most of us, the risk of COVID-19 and of getting a severe infection with COVID-19 is much higher than any risk with the vaccine. And so as physicians, you know, as doctors in general, it's our job to talk to people about risk often, you know, in terms of medicines that we use or vaccines and so on. And generally we choose the ones which, you know, where the benefit outweighs the risk, but there's always a risk benefit ratio. So you take some paracetamol, there's a very small risk that you might get a side effect. You take penicillin for a chest infection, you might get a side effect. And actually the likelihood of getting a side effect with penicillin is much higher than getting a side effect with a vaccine. But the risk balance ratio does change slightly with AstraZeneca when it comes to very young people, because young people are actually not massively at risk of getting serious infection with, and Thomas is more expert in this than me, but are at far less risk than people who are middle-aged or older. And we know that the major risk factor for getting severe COVID-19 infection or dying of COVID-19 or being hospitalized is age. So age outweighs all the other risk factors by masses. It's just a much larger risk factor than anything else, even though, you know, you are at slightly more risk if you're a bit overweight or if you have diabetes and that sort of thing. Age is by far the biggest sort of risk factor, and therefore when you're young, you're not really at that much risk of getting really severe infection. And actually your vaccination is really to protect the older members of the population from, you know, by increasing herd immunity in the population, rather than yourself. And so it's a sort of act of selflessness in a way. But it also it does mean though that we wouldn't want to ask people who are younger to take a risk of a vaccine which might be slightly higher risk than the infection itself causing a problem. And so if you're under 30, that balance starts to just very slightly shift. But we're still talking about something that's like 1 in 100,000 people, you know, so it's just to be clear, you know, your risk of being knocked over by a bus is much higher. Your risk of taking paracetamol and having some sort of reaction to that, or, you know, or penicillin or whatever, is much higher. So the risks are still very low, but then, you know, when we're vaccinating millions of people, that does start to add up a little bit. And if there are vaccines which don't seem to be associated with that risk, then it probably is sensible to offer those vaccines that aren't associated with that albeit very low risk to people who are younger. I guess Thomas and Megan might want to say
very
more.
Comprehensive, thank you. So I think first we'll go to Megan. I especially want to know more about these antibodies that Emma mentioned and how exactly they play a role.
I think Emma thinks I'm much smarter than I am. I don't know very much about platelets at all, never mind these antibodies. I think it's a big gap in our knowledge. We tend to think of platelets as these things that, as you say, when you have a cut, they come along and they help to stop that bleed from happening. But platelets also have lots of immune functions. So it may be a combination that when you have the immune response to the vaccine, you also stimulate the platelets in some way, and this contributes to the effect that is seen in this, as Emma says, very, very small number of people. And so I think we are all very concerned about it, obviously, and there's an awful lot in the media about it just now, but just to remember that that risk to any one particular individual is very, very small. And I think as far as we know, there isn't yet definite evidence that it is the AstraZeneca vaccine that's causing the blood clots, although there are more people than we might expect who've had the vaccine who are having these blood clots.
Okay, yeah, this definitely seems to be something that there's something new happening with it every day, so I guess we'll find out more. One question that we've got, so I think Emma, somebody mentioned that you get blood clots. That's one of the symptoms of having the COVID-19 infection. So is the mechanism of causing the blood clots the same, or are these two distinct kind of normal blood clots and then these more severe ones?
Yeah, they're distinct actually. So the COVID-19 related blood clots are much more standard, I suppose, in a way. So you often, we realized so it's been quite interesting as a sort of doctor looking after people with COVID-19 that we were again taken by surprise. I think this is what happens with a new disease. You learn. And so in the second wave, we knew an awful lot more than we knew in the first wave. And one of the major clinical findings was that people who became very unwell with COVID-19 infection, a large number of them were actually developing blood clots in the lungs. And so we now routinely give preventative treatment to people who are unwell with COVID-19, and I think that's been one of the reasons why our our treatments have substantially improved. There are other reasons as well. We've got better treatments, and we understand the disease substantially more. Unfortunately, the virus has become a little bit nastier to counter that, but we're definitely a lot better at treating it. And one of the things that we do now is to give treatment to prevent clots. But those are standard blood clots, whereas the syndrome that may be associated with vaccination with AstraZeneca is a very specific and rare syndrome associated with low platelets as well, and these antiplatelet factor 4 antibodies.
Right, okay. So the antibodies you mentioned previously, they're to do with the platelets as well.
Yeah, whereas blood clots are actually, we see blood clots quite commonly in hospitals in people, for example, after operations or with any kind of severe illness, including infection. And but COVID-19 definitely has a higher risk of causing it, and there are it's multifactorial, so there will be multiple reasons why people get blood clots with COVID-19, but they appear to be much more of the type of blood clot that we see, commonly in people in hospital, and that we've become much better at managing in patients who are infected.
Great. Okay, so I'm going to come to you now, Thomas. So as we've just heard there, it's all about balancing the risk of the benefit and the risk. And in older people, it seems quite clear that there's definitely more of a benefit of taking the vaccine than this very small risk. But then when we come to younger people, maybe it's not such a easy decision. So maybe you can talk us through that.
Yeah, so there's different elements to vaccination campaigns, and there always are. The first one is protecting individuals. So what is your risk after you get the vaccine of getting disease? But then a lot of diseases spread through populations like COVID has, or don't spread because there's herd immunity. And I think the aim with any disease is to eliminate it, right? So smallpox, we eliminated by vaccinating everyone. That's the only example we've got, unfortunately. But we're close to it with things like polio, where immunization campaigns globally effective, and we're almost there. So there was a question in the chat about what's the point of immunizing younger people if all the older people who might get sick are already immunized.
Yeah.
Which is a valid point. But then there's some relevant points for COVID, I think, specific to COVID. So even though your risk of severe disease is higher as you get older, as Emma said, increasing from teenage years onwards, long COVID, which again, we understand really poorly, but this kind of constellation of symptoms associated with having had COVID infection, that seems to be more common in younger people, particularly kind of young to middle-aged women.
Right.
And the evidence seems to be that the vaccination prevents you against long COVID. So long COVID is really common. I think Emma has published a paper showing that almost a half of people in hospital with COVID develop long COVID symptoms.
Wow.
And then in the general population, maybe 10 or 20% people have long COVID symptoms. So the vaccine in younger people would prevent that. And if we don't have herd immunity by vaccinating everyone, then COVID will just come and spread amongst us. And older people who can't get vaccinated will be vulnerable. The people that Emma was talking about who maybe don't mount such a good response to vaccines might also be vulnerable. And then all the people who haven't had the vaccine will be vulnerable if we get these epidemics every year running through the population. And because I think in Scotland around a fifth of the population are under 18, then if we want to achieve herd immunity, then we need to think seriously about kids and whether to immunize kids. And it's tricky because kids are at really low risk. I'm running a study to look at the actual numbers of children in Scotland who've had COVID and how severe the disease has been. And the numbers are really small, very small risk, but the risk is definitely there. I don't know how you compare it to Emma, perhaps for meningococcus, which again is a serious complication, but also quite rare. We vaccinate for that.
Okay.
So you could argue it's very unlikely to affect kids, but it can affect them seriously. It's worth vaccinating for that reason.
Right.
We could say, like we do for flu, every year we vaccinate toddlers and primary school children for influenza. And we're not very explicit about it, but the information leaflets given to each parent says the main reason we're doing this is not to protect your child, to protect your child's parents and grandparents and teachers. And the children are being vaccinated for the greater good. And I think that will be a discussion that we need to have about COVID. And the AstraZeneca vaccine seems to have these very, very rare side effects, which might affect if you're a parent and there was this risk of a very, very rare complication, I think parents will be put off more than they would do for their own decision. But there's other vaccines out there, the Pfizer vaccine, the Moderna vaccine, which have been trialed in kids, and so far don't show these signals of a rare side effect. And we need to have a discussion with parents teenagers and say, these are the benefits to you directly, these are benefits to society. Do we think that this is something that we should invest in? But like any question, the more data, the better. And we don't have the data right now, either for the safety of the vaccine in younger people and also the burden of disease. I don't know if Emma or Megan have got anything to add to that.
I'm getting shakes of the head. So I just wanted to pick up on one of the things that you said there about the flu vaccine. So is that because the flu vaccine and potentially COVID is more transmissible among young people? I'm just kind of thinking of like toddlers licking things and just generally the school near me, all of the young people are, you know, jumping on each other and having a great time, which is obviously lovely to see, but it does make you think that a virus will be having a field day.
So that's partly true for flu. Schools seem to amplify flu pandemics. So if you close a school during a flu pandemic, you will interrupt transmission. Okay. And that's because younger people play a big role in transmission. That doesn't seem to be so clear for COVID. We used to think that they didn't transmit disease at all, but they clearly do. But I don't think they serve to amplify disease in the same way that flu does. So I don't know if Emma has any more information than I do, but certainly if you look at the Scottish data, there's not been big outbreaks in primary school, for example. So there's been 1 or 2 cases in the class, but there haven't been cases really where the whole class gets COVID, which is what you'd expect if it was as transmissible as flu, for example.
Okay, so there's definitely a difference there. So we've got a question coming in, and it kind of relates back to what Megan was saying about our peak immunity being as a teenager. So I guess if we vaccinated when you're younger, would that mean that you have a greater immune response? And could that be then a benefit going forward as you age because you had it at a younger age? Or is that not really how that works?
I guess historically, I would have said it's better to get a vaccine when you're younger because certainly past about 50, 60, 70, your immune system to a vaccine is not going to be as good as it was when you were young. And we mostly vaccinate young people, partly because they're in the healthcare system much more readily and to obviously protect them against childhood diseases. And then we often vaccinate people at the other end of the spectrum because they're now more at risk of infectious disease. With some vaccines and many diseases, if you have that when you're young, you'll have lifelong immunity. The negative to that is things like the tetanus vaccine, which many of you will have had multiple times because it's not a great vaccine, so we have to keep giving it again and again to boost you. I think what we can see with the mRNA vaccines in particular is that they give this really strong immune response, even in the elderly. So in those cases, if a new virus comes along, we can make a new vaccine and we can protect the whole population, providing they prove to be safe in children too. And it wouldn't be as important that you had the vaccine when you're young. What we've yet to see with the mRNA vaccine is just how long the antibody and the T-cell response lasts. And I think we'll all be keen to see those papers in the next 2 or 3 years to see how much there is a drop and how long the protection is given. And that's a big unknown at the moment. So I would say, yes, it's good to be vaccinated when you're young, but hopefully with these new vaccines, we can also really protect elderly people as well.
Okay, so kind of going back a little step there. So I think Emma made it quite clear that the reason that we didn't see these extremely rare blood clot syndromes in the trials was because the trials were, even though 30,000 people seems like a lot, when we're talking about something very, very rare, we're not going to pick it up until it goes into the millions, which is what it has now. So a question that we've had come in is that, is it because the AstraZeneca vaccine has been given more? It was like one of the earlier ones. So is there a chance that as more and more of the other types of vaccines are given, that we might see a similar thing happen? Is that a very, very rare thing comes up? How likely is that?
We've given a lot of Pfizer, for example, as well. So there's been a huge rollout of Pfizer and Moderna in the US as well, vaccines, which are the mRNA vaccines. And so yes, it has been given on the same sort of, these have been given on a similar scale to the AstraZeneca. And so it's sort of possible to compare data sets. I think it's quite important, though, to be a bit cautious because you're looking at different populations, and then there are different variants of the virus in different countries and so on as well. But yes, these vaccines have been used at very large scale as well. And in fact, we've used a lot of Pfizer in the UK. So healthcare workers have had it, predominantly have had Pfizer, for example. And we're not seeing the same level of safety signal with Pfizer or Moderna at the moment. But I mean, of course, this is active. We're doing very active sort of data collection, and things can change over time, and we may learn more. I think it's very likely that this is going to be some sort of rare genetic thing, probably, that some people are more susceptible to that type of problem. And I suspect strongly that we will learn about that over the next coming months. There'll be a Nature paper that tells us about the mechanism of the anti-PL4 antibodies, and it's quite likely to be genetic. We know that there are genetic associations, for example, with severe COVID-19 as well now, and so we're going to learn an awful lot in the coming months. Maybe you should hold this again in 6 months. So we'll have more answers.
You in now. That's a really interesting point then. So you said that this is an if, but if it transpires that there is some sort of genetic risk factor, then maybe we could screen people and say, okay, you are at higher risk of developing this rare blood clot syndrome, so we'll give you one of the other vaccines.
I think in the future we're all going to we may all be offered to have our genetic, our genome done. It can be done quite cheaply already.
Yeah. That is a whole nother panel discussion.
Yeah.
So I'm gonna
but then you've got to know what to do with the results.
As interesting as I would love as a geneticist to go down that route, I feel like that is a completely different Q&A session. Okay, so I've got a question which I feel like we touched upon, but just maybe a little bit of clarification. So the very rare blood clots are characterized by blood clots, but also low platelet. And as you just explained, the platelets cause the blood clots. So how can that happen? Or is it that we just don't know that yet?
Blood clots are complex, so it's not just platelets that are involved in blood clotting. Those are one of the cell types that are involved, and so it's slightly more complicated than that. In that there are there are proteins and things that are involved. But my guess is, and this is a guess, and I don't know the answer. Maybe one of the other panelists does. It may not. It probably isn't known. But my guess is that the platelets are actually migrating to where the clot is, so there will probably be an awful lot of platelets sitting in that blood clot, and that have come out of the bloodstream and into the cavernous sinus, for example, or wherever the blood clot is, and so you detect a lot lower, a lot fewer of them in the bloodstream. That would be the most likely explanation, I think. But there could be other more complicated reasons that again, ask us in 6 months. I think we'll probably know more. But yeah, I mean, we see it quite often with immune cells, and this is Megan's expertise, but immune cells track to where the infection is. And so sometimes you can pick up like low levels of other types of cell in the bloodstream, but actually they've all migrated to wherever there's an infection, for example.
Yeah, Emma's very much right there, that we do know in the case of immune cells, and this happened in COVID, that the numbers of your T cells in particular are low in the blood early in the infection. And that's not because suddenly all your T cells were wiped out, but just that they had moved to where the virus was to try and deal with it. And so, yes, I think we're all speculating now. It could be that if you're measuring low levels of platelets in the blood, it's because they've been activated and are tracking to one particular site where they think they're doing a good job, but actually they're causing disease.
Okay, that makes a lot of sense. So you're measuring in one area, or in the blood, but actually they could just be that they're somewhere else. That makes a lot of sense. We've also got another question, which I feel like the answer is going to be we don't know yet, but I'm just going to ask you anyway, is there any evidence that this is more likely to happen if you've got a previous history of blood clots?
I'm looking at the clinicians.
I think I can just add a very general point, is that there's really few cases, and these are all cases being reported either to the MHRA, which is the UK agency, or the EMA, which is the European agency, and that's just cases of clinicians reporting cases, and then they collate them together and they look at the data. But because of concerns about patient confidentiality, which you can understand with this very rare condition, you might be able to find out someone's medical, they're not being released in a detailed way, just the aggregate analyses. So we're all looking at the same report. That we don't have access to the raw data. But what people are saying is that there doesn't seem to be any consistent risk factor that they can identify.
Okay.
So the EMA has looked at more cases of the cerebral venous sinus thrombosis, which is this rare complication, I think over 100 now. And they can't find any particular risk factor in terms of age or medications or these other kind of factors that I've seen in the chats. There doesn't seem to be any clear predisposing factor. I think, as Emma says, it sounds like it's individuals who are predisposed and so
obvious things that we might have thought of that might have been a risk factor have kind of been ruled out so far, and we're kind of looking for more evidence at the moment.
Yeah, I think that's right, and it seems there hasn't been a signal that way, so you might have expected it, but there hasn't been. So it does look like a completely different syndrome from the kind of usual blood clots and the usual risk factors for those.
Okay, yeah, I think we kind of discussed that earlier, so it makes sense that it wouldn't necessarily be linked because they're quite different situations. Okay, so I'm gonna come back to something that we sort of touched on earlier, but has come up in the chat. So I think we all remember that when the vaccines were first approved, I can't even remember which way round it is, which is bad, but one of them was supposed to be 3 weeks between doses and the other one was 12 weeks, something like that. But obviously it's not really what's happened, because we've got X number of million people. I think I saw on the news it's like 50% of the population now have got their first dose, but are waiting much longer than 12 weeks or 3 weeks to get their second dose. So I guess I just wonder if any of you can comment on why the government decided to make that call and if it's a medically sound call.
Emma, do you want me to start?
Go for it.
From an immunology perspective, what happens when you have an immune response is that your T and B cells increase in number to try and fight the virus, or in this case, to make a protective response with the vaccine. And then they decline again, and that's totally normal. And there is some evidence that if you hit your cells in that declining phase, they won't make a good immune response again. So that you want to wait at least 3 weeks before you get your booster, so that they have a bit of a rest and then are ready to respond again. And there's a little bit of evidence from some basic immunology studies that waiting a bit longer is actually better, because your cells have had a bit more of a rest, so they've got a bit more energy and are ready to go again. So from an immunological standpoint, I wasn't too concerned when I heard that they were going to increase the length of time between the two doses. And my understanding of the government's position was that the number of infectious cases in the UK had increased so much that the risk to the larger population was much greater than the benefit to the individuals getting their second dose. The first dose gives you a large immune response, and you will get more of an immune response after the booster, but the biggest increase is from that first dose of the vaccine. Okay. So those people were pretty well protected, And we wanted to protect as many people as possible because of the high level of the virus at that point that they made that decision.
Okay, so it was a bit of a political decision, you know, health policy, but also from an immunological standpoint, it's actually the shorter time is the problem, that you don't want to go less than 3 weeks than actually go in longer. That's really interesting. Okay, Thomas, Emma, did you have anything that you wanted to add to that?
I'll just add, I think there's a lot of debate at the time about this amongst health professionals too, and amongst doctors. And I think the BMA came out against changing the schedule. But in retrospect, I think it's turned out to be a very sensible decision. We've made the vaccines reach twice as many people. We're already seeing the effects of the vaccine. And as Megan says, the effect of the first dose has been really substantial, more than people expected, I think. And the protection from the first dose has also been really high. In Israel, they did studies looking at the protection after the first dose, and that seemed to be much more effective than people thought it might be. So with the benefit of time, it turns out to be, I think, a good decision.
I agree with that. I was a little concerned about the possibility that you might have a kind of partial immune response, and that it might actually make it more likely that new variants might circulate more easily, but that hasn't come to pass. And I think it was a very important decision, a really difficult decision that was made by a very expert group, some of whom I'm lucky enough to know, and I think they made the right call, and I think it was very difficult, and it wouldn't be something that we would do standardly. So I think the BMA, I can understand the objections that, you know, you want to give medicines as trialed, and that it's quite rare to deviate from that. But there was there was evidence with the Oxford vaccine that the delayed people who had delayed vaccination actually had stronger immune responses, and so there has been some evidence with that vaccine that delaying was useful, but that's not the case with Pfizer and so on. That certainly wasn't available. That evidence wasn't available, and so it was a decision that was made in a kind of non-peacetime, you know, a sort of wartime decision. And I think I agree. I think it was a really good decision. I think it was quite a brave decision, and it required the expertise of scientists and medics who've spent a lot of their careers thinking about these things, and I'm kind of grateful to them. I think they made the right call on it, but I don't think it was an easy one.
Yeah, I guess that's something that we kind of forget. We see the politicians standing up and making these announcements, but actually it's not them necessarily making these calls. It's people who have really spent a lot of time thinking about it. And as you say, a very difficult and sort of unusual decision, but a very unusual circumstance it's being made in as well. Okay, so that's great. So I'm just checking the question list. Things have moved around a little bit. Okay, so we've got a question about, say you have COVID-19. Obviously, if you have symptoms or have been contacted by test and trace, then you should be isolating at home. But if you're non-symptomatic, you don't know you have it, and you get the vaccine whilst you are infectious, what would be going on with your immune system? Could that be a problem? Would that actually be helpful? How would that kind of work?
Well, I start with that one. Emma's nodding. It's likely not to have a massive negative impact on you. I think the worst that might happen is that you don't make quite as good an immune response to the vaccine, because you've already got some of those T cells and B cells and antibodies floating around in your system, and they may limit the amount of immune response that happens as a consequence of the vaccine. But I don't think we have any data that tells us that that would be the case. That's just a speculation of what might happen if you vaccinated during an infection. Okay. We sometimes, somebody asked me this the other day, and the only correlate that I could think about was older individuals who have shingles, which is a reactivation of the chickenpox virus. And those individuals are sometimes treated with the chickenpox vaccine to stimulate their immune system. Okay. So that is something we have evidence on, and we know that that leads to a bigger immune response and helps them control the resurgence of the chickenpox virus, so their shingles sometimes reduces more quickly.
Okay.
So I think people shouldn't, as you say, if they think they have COVID-19, they should get tested and they should isolate. But if it were to happen that they were vaccinated while they were infected, I don't think that would be a serious consequence to them. And then they would get their second vaccine and they would get a booster immune response. So they would be protected in the long term as well.
Great. Okay, that's great. Anything you wanted to add, Emma and Thomas? No? Okay. Right, so we've got a question here about kind of risk factors. We've spoken specifically about these very rare blood clot syndrome, but just generally about side effects from the vaccine. Is there any evidence that gender, ethnicity, or any other sorts of risk factors would increase or decrease your risk of having side effects from the vaccine? Emma's nodding.
Sorry, I missed the beginning of that, but there was a question. Basically, the question is, are there any risk factors which might make you more likely to have side effects from the vaccine? Is that right?
Exactly that, yeah.
I think the clearest one is probably preceding infection, being a kind of risk factor for slightly more symptoms, but not not strongly. It's not. I don't think there are other very strong links to other issues. Unlike so people have sort of asked about gender and ethnicity. I'm not sure if that was with regard, for example, to blood clots. So we know that females are more likely to be at risk, for example, and both from, I don't know what that relates to, but from COVID-19 disease itself and also to the blood clots. But men are much more likely to get severe disease with COVID-19 overall, and much more likely to be hospitalized with the infection. And there is there's a difference between the sexes that way, which is quite interesting. But I'm not aware of anything popping up in the vaccine trials in terms of side effects from the vaccine, for example, relating to being female or male.
Okay.
You're probably slightly less likely to be symptomatic if you're a bit older.
Okay.
Sorry, Thomas, I was going to add something.
I was just going to say exactly that. And I think here we're talking about the side effects that we'll all get from the vaccine in terms of feeling a bit fluey or achy or a bit of a temperature afterwards. We're not talking about the risk of severe side effects. There doesn't seem to be any particular risk factors for severe side effects for any of the vaccines. But the studies have shown that.
Yeah. Sorry, I was just going to say that when I got mine, I was really glad to feel a little bit tired and achy, because I was just thinking, this is great. My immune system is ramping up.
My dad, who I think is on the call, got very ill after his, and I said to him, "That's great. It means it was working," which I'm not sure he appreciated. But yeah, I definitely see that logic.
But we should point out, even if you don't feel that flu sickness, you still are likely to make an immune response. The two are not absolutely directly correlated.
Yeah, absolutely.
Okay, great. So kind of coming back to something we talked about earlier, I think, Thomas, you mentioned smallpox, which we all learn about Edward Jenner in school and inoculation. So we've globally eradicated smallpox. Is that something that could happen for COVID-19, SARS-CoV-2? And there's a big question. I'm putting a disclaimer to say that obviously nobody has the answer for this, but for your opinions, what do you think?
I'm happy to start, and I guess I'm a little bit pessimistic about this. Smallpox was easy. Smallpox was not easy to eradicate, but it was a virus that didn't change very much because it's a different type of virus to the COVID-19 virus. And also it only infected humans. So we only had to worry about the human population. The really far opposite of that is an example is influenza virus, which circulates in lots of different types of animals. People will have heard of swine flu and bird flu. So I think we will never, ever eradicate influenza virus, for example. COVID-19, the SARS-CoV-2 virus, we do know can infect other animals. So there is a potential reservoir outside the human population that we would have to also deal with if we wanted to eradicate it completely. And I think that the global levels of infection will make it incredibly hard to completely eradicate in the near future. Whether it's possible in the much, much longer term, I am pessimistic about, but could be possible.
Okay. Emma, Thomas, do you want to add anything?
I think, yeah, it's clearly a long-term project. I read somewhere that if the current rate of vaccination continues, it'll take 5 years to vaccinate everyone in the world. And that's assuming we vaccinate younger people and we can vaccinate people in poorer countries. It's kind of it's not in the near future a question that we can answer. But I mean, in the UK, the lower we can get the levels, the better, because the more people immunised, the less chance there'll be outbreaks of our life being interrupted again.
Yeah.
And my personal opinion is that unless we immunise kids, we'll be potentially in a situation where we have these school outbreaks that we don't know what to do with, because clearly it's not harmful for kids. But are we going to close the school because there's COVID, or are we going to keep the schools open and then accept that we're giving COVID to everyone in the school? I don't think we've got a framework yet in our minds for how to deal with that. But
yeah, it's a tricky call.
We've got time to discuss that. I'd be interested to see what Emma and Megan think about schools and children, because speaking as a parent, the interruption to your working life from COVID is huge in terms of having to quarantine, whole classes having to quarantine, staff members being off. Yeah, I don't know what we do in a situation where everyone's safe, but the children are still potentially getting COVID. Or whether it could be that all the adult population is enough to restrict transmission and we have herd immunity, but I'd be interested to see what Emma and Megan think.
I think we're probably stuck with this virus for the moment as well. And of course, it's possible that we might eradicate it. I think we had the opportunity to eradicate it earlier. And I think we can learn from this. So we do need to get more responsive about pandemic viruses. And somebody asked, I kind of noticed in the chat a bit earlier that someone asked, are we going to be seeing this sort of thing more often? I think the answer is yes. We live in a very interconnected world, and it's like the continents have kind of closed in together because we fly all over the place all the time. And we know, so Thomas and I were involved in writing papers where we've looked at the genetic sequence data to understand more about how the virus sort of transmits and so on, and also how it works. And one of the things that we found from the genetic data is that the virus was introduced more than 1,000 times into the UK in the first month of the outbreak. And in Scotland, for example, that was probably around 300 times at least. And that will be an underestimate because that was based on the genetic sequencing that we did, and we didn't do it for every single person. So, that will be an underestimate. And we know that we imported it that many times. And then in the second wave, we actually, most of the lineages that we had in Scotland, for example, became extinct. But a couple of them were persistent, but we re-imported them all again through travel. And so importation from travel, international travel, is really a really important route of transmission. And I mean, I've worked as a consultant in Glasgow for about 10 years, and we've seen many imported nasty viruses like Ebola virus, Crimean Congo hemorrhagic fever, Seoul hantavirus, and things like that. And I don't think that my predecessors, experienced that in the same way. These sort of viruses that might be more associated with tropical infections are causing larger problems. We know that from the Ebola outbreaks that we've seen in Congo and also in West Africa. And so, yes, I think we have to be prepared, and we have to be prepared to stamp down on them faster. And think very carefully about how best to, we need to invest in pandemic preparedness and vaccines and new treatments.
Yeah, definitely.
I couldn't agree more with Emma. I think, yeah, we took her off the ball with coronavirus, and then in some ways we were lucky because the mortality rate is relatively low, maybe 0.5% to 1%, but if that were to go up to 2 or 3%, or if it were to affect younger people more, we'd be in a really tricky situation.
Even worse. So I was going to kind of come to this at the end, but since you've broached it, we'll come to it now and loop back. So I guess at the beginning of the pandemic, on social media, lots of people comparing to the Spanish flu, or as we'll call it, the 1918 influenza pandemic. So I guess I kind of maybe I can guess the answers from what Emma's just said, but why now? Like you just said, we've got people like Ebola, we all saw it on the news, was awful in certain places in Africa. As you just said, some people had it here, but it didn't spread in the same way where everyone just had it. So, you know, why this virus? Why has this virus caused this pandemic? Similar to, I guess, the last one we had was in 1918, because we've been having air travel for, say, 50 years. Obviously, it's increased recently, but yeah, I guess why is this a pandemic?
I'm looking at Emma as the more virologist, but I can make a start on answering that question. I guess I wouldn't go quite so far back to 1918, but would remind everyone about SARS and MERS, which were coronaviruses that caused infectious disease, mostly in the Far East and also in the Middle East. And we were very concerned about those, but they didn't spread. Partly that was because the countries managed to control them quite well, but also because the virus didn't transmit very well between people.
Okay.
So although there may have been groups of people, the virus wasn't able to get between them. And so if it can't get between people very well, then those individuals are not going to go on and spread the virus to more people. And I think I'm right in saying that MERS was a much more severe infection. So those individuals were more quickly in hospital and then less likely to spread it to others. Whereas with SARS-CoV-2, a lot of us who get infected are asymptomatic. So we walk about our lives transmitting it to other people. And also it's a much more transmissible virus than the original SARS and also MERS. So it's a little bit about the consequence to the host and also about the virus. And I think, as Emma says, we live in this very interconnected world. And once it was out in the population and starting to mutate, then it was just popping all over the world through air travel and congregations of people being together, and that easy transmission of the virus between individuals.
I could add something. I think Megan's covered the biology really well. I think one thing that as humans we've got to really think about is that we're intruding on the ecosystems that we haven't previously intruded on. And so we know that SARS-CoV-2 was lying in wait for us in bats. And we also know that there are many other SARS viruses in bats that look like they can also infect human cells but haven't yet caused pandemics. And I think that we have to think about how we interact with the world. And I mean, I have views on that. It's not my area of expertise. I'm not someone who works on biodiversity, but we are seeing these problems because there are viruses in other animals, particularly bats, for example, and we are intruding on their territory by population growth, and that increases the risk of viruses jumping from animals into humans, and we've seen it many times. This is what happened with HIV as well. It jumped many times from chimpanzees, and in one case at least from a gorilla into humans, and that was because of changes in our social sort of demographics. And you know, there's some excellent books on that about there's one by Jacques Pepin called The Origin of AIDS, which goes into a lot of detail about how the way we live our lives it can cause the risk of infection. And I think live animal markets where you have lots of animals housed together alive in cages where they're in very close proximity to each other is a risk, and something that we should look at as the human species. And I also think that we are destroying large amounts of forests, which is bad for the planet. And but it also it puts us much more in contact with species that we wouldn't normally have been in contact with, and increases the risk of pandemic viruses. And somehow we have to think about how to behave more responsibly collectively. And that's something that I would hope that perhaps our politicians of the future might start thinking in the longer term.
Mm-hmm. So I guess in conclusion to that question is that actually comparing it to the 19 is maybe a slightly blinkered view of things, and there's actually been lots of transmission of new viruses that have spread. It's just that a unique set of circumstances to do with the transmission being higher and the mortality being lower that meant that this one spread around the whole world. But we should definitely be on the ready for the next.
It's quite likely there have been other jumps, but they haven't spread. You know,
right.
Like this may have happened before, and maybe a couple of people got really sick and then they didn't transmit it.
Yeah.
But yeah, that unique set of circumstances resulted in this pandemic.
Okay, so I've just got a couple of more questions which are actually linked to the kind of more longer term thing. So one of the questions that we've got is, with COVID, the SARS-CoV-2 virus mutating, is it possible that it will become less dangerous over time, more like cold or flu and less people dying, or is that not really likely to happen? Obviously we can't predict what the mutations will be, but in terms of probabilities.
I'm not an expert, but the last year has not given us any hope that that's the case. We've only seen variants which are more transmissible and more deadly. There were a lot of predictions at the start of the pandemic that all viruses become less lethal, perhaps as they evolve in the host and adapt to their host. But for the last year, I don't know if Emma and Megan agree, there's no evidence to support that in the data we've seen.
Yeah, I agree. I mean, I've been really disappointed. I was brought up by people that taught me that viruses become less lethal with time, and that we've seen very clear evidence that this virus has become a little bit worse, not massively worse, but a little bit worse. And of course, over the period of 100 or 200 years, that might be quite different. But in our lifetimes, I'm not sure that we're going to see that this virus is going to get milder, but we will be more able to deal with it because we'll have vaccines, and so it becomes less important because of that.
If you kind of think about what's good for the virus, all it wants is to replicate and get into new people. Because it can cause asymptomatic and mild infections in many people, there's no advantage to it to cause a milder disease in some individuals, because it can easily get around the human population without having to worry about becoming less deadly to us. So viruses only change to have things that improve their ability to transmit or to infect.
Yeah, that makes sense. That makes sense. Okay, I've just got a couple more questions before we wrap up. So one of them is kind of linked to that idea of like comparing COVID-19 to the cold and the flu. If we are having updated vaccines potentially every year that are responding to the changes that the virus is making and the variants that are circulating, would that mean that we would have antibodies against multiple variants, or would we only have the antibodies for the most updated one and potentially give chance for a previous variant to come in? How exactly does that work?
I can start with that answer from an immunology point of view. Your immune system is amazing, so it can pretty much take anything you throw at it. So if you look at, if you were to take my blood, for example, and look at how many different influenza viruses I'd responded to, you would see that my immune system has done a pretty good job every 2 or 3 years boosting my immune response against the different flu viruses that circulate. And that's because we have these amazing cells that can recognize these very small differences sometimes, or larger differences that happen between the viruses. And we can store those memories throughout our entire life. So if you have been exposed to the original version of COVID-19 virus, SARS-CoV-2, and then get vaccinated with an updated vaccine, then you will keep your old memories and you'll make new memories to that vaccine. So for most people, you will store that memory. We don't really know yet quite how long the antibody levels will last. I think we were very concerned this time last year that the antibody responses to natural infection were waning very, very quickly. But more recent data has shown that certainly 6 months after an infection, you still maintain a good level of antibody against the virus. And again, we'll need to wait and see, but we're hopeful that the newer vaccines will also give us a long duration of an antibody response.
That seems pretty comprehensively answered. Is there anything Emma or Thomas wants to add to that? No, great, okay. And yeah, I think that is pretty much all of the questions that I have at the moment. So I've noticed a couple of questions have come in on the chat, which we absolutely definitely covered earlier on. So I'm not sure if you missed it or you came late. So just to let everybody know on the call that we are recording this call, and it will be put onto the ScotSci YouTube channel, which you can just search ScotSci on YouTube, and everything, the whole chat will be on there. We've also got some other previous discussions that we've done with other scientists on different topics, so check that out for your questions. And then maybe one last one. So Emma, you touched on the fact of us, going more into the environment, coming into contact with new things. I mean, as you said, overpopulation and growing industrialization around the world is obviously bringing us into closer contact. Will we ever know where exactly the SARS-CoV-2 virus came from? And I guess maybe on that point, does it matter where it came from for the future? If we know where the other ones are, just maybe to kind of finish on that point, if you...
Yeah, I mean, I think we're very clear that it came from bats. It may have come through an intermediate species, in the small animal market, or it may have come directly from bats. And we probably will know more because there's quite a lot of work going on to look at bats, particularly, and other animals in China. So it undoubtedly came from there originally, and there's research based on the genetics showing that. So you can use the genetic signal as a kind of clock. That it probably has been sitting in bats for at least sort of 50 years or so, ready to make the jump. What we don't know is, so I'm not sure if that question was around something that President Trump said that it came from a Chinese laboratory or whatever, and I think there's no evidence for that at all. It doesn't look like an engineered virus, so it doesn't look like something that a human has engineered. It looks like something which has originated naturally. It still could be possible that it came, in fact, people were using samples or something, or in contact. It's obvious that it came from contact with animals, and the highest risk and the most so in labs, people wear protective equipment and so on, and the likelihood of a transmission occurring in that environment isn't very high. But there are people in live animal markets where there's live animals in very close contact with each other, potentially with high viral loads, and people kind of contacting them very directly. So that is very much more likely to be the risk. But we certainly know they originated from bats. There are a lot of viruses that have made the jump from bats into humans, actually. Bats are very diverse, so they're not just like one type of animal. There are many, but they do harbor a lot of viruses, and so don't pick up a bat if you find it at home. Don't kiss it, and don't chop it up. But a lot of viruses seem to make the jump that way from bats into humans. The laboratory question, there's just no evidence for it. It doesn't mean that it couldn't have happened. It could have happened, but there's definitely no evidence that it did happen. And it certainly doesn't look like an engineered virus, as in something that someone was playing about with and changed genes and things and to make it infectious to humans. That doesn't look likely at all.
Okay. Megan, Thomas, have you got anything that you'd like to add on that front?
I think Emma's advice on not picking up bats is excellent.
Okay, good. Okay, so I think we'll wrap that up there. So first of all, thank you so much to our panelists. It's been really great to chat to you and get your insight and experience on a whole range of topics that I've put to you this evening. So thank you so much for giving up your time and coming and speaking to us this evening. And I'd also like to say thank you very much to all of our audience members for coming along. It's been some great questions, and I think it's really good to have conversation about what's going on and get some answers on these questions. I've had a request to follow up on the suggestion that we hold a similar event in a few months' time when there's new evidence. So I will definitely be keeping an eye on things, and maybe we will put up a new event, and I'll be asking you guys to join me again. So keep an eye on the Scots Eye website where we put all our new events. So at that point, I think I'll just say thank you very much to everybody for joining. And as I mentioned, the video will be onto our YouTube channel very soon, so you can rewatch, you can share it with your friends. And yeah, I'd just like to thank everybody again.
Thank you, Kathryn, for hosting.
Thanks for the invitation and for all the questions.
Thanks for the invitation, and really nice to hear everyone's questions, everyone's thoughts. That was great.
Yeah, definitely.
Great, thank you very much. Bye.
Okay. Bye.